So, good morning, everybody, or good evening, or good afternoon, depends on where you are in the world. Today, I will, the Manual Management Network will host a small webinar about reliable data from measurements of ammonia emissions, which is needed for the purpose of developing methods to reduce emissions and calculate emission inventories. And therefore, we very much appreciate the three researchers with much experience in measuring gas emissions from farming and how to use the chamber and tracer measuring techniques, who today will give a presentation about these methods. I will just add that we are also, the Manual Management Network will in April held a seminar where scientists present how emission measured with these techniques can be used to develop greenhouse gas emission models. So with these words, I will say, I'll just present myself. I'm Sven Sommer. I'm co-chair of the Manual Management Network and professor of the environmental friendly farming at the University of Aarhus. So the first speaker I'll give the screen to is Dr. Johanna Petersen, who will present using dynamic flux chambers for measurements of ammonia emissions from land-applied manure, accuracy of relative effects. Johanna is employed at the Department of Biological and Chemical Engineering, Environmental Engineering Group, Aarhus University. And she has much experience in measuring emissions using the dynamic flux chamber technology and has specialized in high time resolution measurements of ammonia and non-methane volatile organic components in the field after field application of liquid manure. Johanna, the word and the screen is yours.
Thank you for the introduction, Sven. I will share my slides. I believe you see my slide in presentation mode now. If not, please let me know.
Yes, we do.
Yes, thank you, Sven. So Sven already introduced what I'm going to talk about. So here's an outline. I'll start with a brief introduction and also introduce a little bit about ammonia emissions after field application of organic fertilizers, as I realized that most people in this online room might be working with the greenhouse gases. Then I'll introduce the concept of dynamic flux chambers. And then I'll spend quite some time with talking about different sources of error when we do field measurements and how we can try to validate our measurements, as this is quite important. And then I'll briefly talk about the importance of auxiliary data, as well as briefly describe a model that we use for estimates of these ammonia emissions. And in the end, if there's time, I will come with some examples of recent research we have done. So first of all, I think we can all agree that accurate measurements are very, very important, but why? Well, of course, we need accurate measurements to support climate actions, to do inventories. But a really important thing for me is that we all agree that we have to mitigate the emissions and reduce them. So we need new technologies and new treatments. And in order to validate these, to actually get them out in the world and working, we need to have good, reliable measurements to ensure that they are indeed reducing the emissions. So I'm working with ammonia primarily, but also volatile organic compounds. And I just want to mention that the ammonia is an indirect source of nitrous oxide. So it is also an indirect greenhouse gas. Furthermore, for the ones who are agriculturally interested, ammonia emissions is a really large nutrient loss for the farmer. So in a quantity of nitrogen, nitrous oxide emissions is not that big a loss, but ammonia can be a really large economical loss and also cause yield losses. So therefore, it's important that we can reduce them. So I have primarily worked with organic manures, but the measurement techniques are the same for inorganic manures, so mineral fertilizers. In Denmark, the sources of organic manures is pig slurry and cattle slurry from the agricultural sector. And then we have a lot of slurry digestate. So we have a big production of biogas, where slurry is used being anaerobically digested, and the effluent from this is then field applied in the same way as slurries. The sketch in the middle is, I'll not go into detail with it, I'll come back to it a little bit later because it's from this ALFAM2 model. But it's just to show that when we field apply organic fertilizers, you can roughly say that it's divided into a fast and a slow pool. The slow pool is what infiltrates into the soil, and from here we'll only have really low emissions. And then we have a fast pool, that's really what's left on the soil surface, which can have high emission potentials. So when we're out in the field, there's a lot of different factors that affects the ammonia emissions. We have slurry, physical and chemical characteristics, where the most important ones is the dry matter content and pH of the slurry. Then we have a lot of soil parameters, and the climate with high temperatures and high wind speeds will increase emissions, and rain can suppress it. And then we have management choices as well. For example, how do we apply the manure out in the field? And just to complicate things, there's interactions. So there's a lot of things that has to be investigated and taken into account when we do experiments where we want to kind of investigate if a certain treatment can reduce emissions. When we talk about measuring ammonia emissions after field application, we can roughly divide the techniques into two categories. The first one I want to mention here is the micrometeorological measurements. That is absolutely great for estimates of absolute emissions because we do not, it's a non-invasive measurement, so we do not manipulate the emitting source. But there is some challenges with these methods. For example, you cannot have obstacles like a tree or building close by. You need larger homogeneous field areas. And then maybe one of the biggest challenges is that you cannot have other emitting sources close to the field or the plot that you want to measure. This means that it can be really, really challenging to have replicates and test more treatments. And this is where the dynamic flux chambers is great. You can see below there's 3 different pictures of 3 different styles of dynamic flux chambers. The ones to the right and to the left are both kind of wind tunnel style, where the one in the middle is not shaped like a wind tunnels, but all 3 are dynamic flux chambers. Here we put a chamber on top of the area where we want to measure the emissions. So we manipulate the area. It's not a non-invasive method. Therefore, we cannot use it to estimate absolute emissions. But we can use it to test relative effects of treatments and, for example, application techniques. The advantage is really that it's a small plot area, so we can have replicates and thereby make a better direct comparison. So what is a dynamic flux chamber? I would say that it's you have a chamber you put in an area that's well defined, and then you have an inflow and an outflow. In the inflow there, that's the blue points. You need to measure the concentration. Then you have a flow of air over the soil surface, and then you have to measure the concentration of the air leaving the chamber again. That's the red points in these cases. Then you can use the concentration differences between these 2 points, as well as the knowledge of the airflow in the chamber and the area to calculate an emission flux. So it's important that we can measure the concentration of ammonia, and this we can also, again, roughly divide into 2 categories. We have the measurements that are integrated over time, and here a very used method is acid impingers, where the airflow is bubbled through an acidic solution that will capture the ammonia. Every time you want a data point here, you need to go out in the field and manually change the acid. What I have been working a lot with is a high time resolution measurement. In this case, there's a picture of a cavity ring down spectrometer, where we have continuous measurements. So that enables us to have high time resolution because we don't have to go out in the field to sample every time we want a data point. And these are examples of data. To your left, you can see examples of time integrated sampling. You see fewer data points, a little bit more in the beginning where you usually sample more often. And then in the end, you don't sample that often. And to your right, we see the high time resolution data, or an example. And I don't want to go into the details here with the treatments. This is really just to show you that we can capture more details with the high time resolution data. And maybe use that to explain why some treatments can mitigate emissions in some cases, but not in others. However, it's really, really important to emphasize that if you're interested in cumulative emissions, then both methods are equally good. So, sources of error. Why is this important? Well, we have a lot of sources of error out in the field, and if we are not aware of them and take them into account, it can really mess with our data. So one thing that's important is, of course, the slurry that we apply. We need to know the concentration of the total ammoniacal nitrogen quite well, because usually we express the emission factors as a percentage of this. So if that analysis is wrong, your result will be wrong. Then also something very low practical is the application rate in the whatever machinery you use to apply the manure. That machinery should also be calibrated in the same way as you would calibrate any instrument in the laboratory. So you know exactly the rate of slurry leaving the tractor. Then we, of course, have the measurement system itself. We have some airflows, both in the chamber and in potential sampling tubes. We need to really know what the airflow is and avoid leaks. And it's also really, really important to do a good quantification of the background. And then, of course, the concentration measurements themselves, and this is both manual handling, laboratory analysis, and for the online measurements, it's really, really important to calibrate to be sure that it measures well and accurately. So I mentioned before that the airflows in the chamber is very important. And one thing we really want to have is kind of a homogeneous turbulence over the soil surface within the chamber to avoid to have dead spots or hot spots. I have used two different methods to investigate this. One is the computational fluid dynamics. These are the round circles. That's the soil surface within a chamber. So you can see we have a big difference here depending on what airflow we have. To your left, it's kind of blue all over. So we have a homogeneous turbulence, where to the right, you see some yellow big points in the middle, and that's where we have a high turbulence. So this could create unrealistic emission conditions. A more simple way to test this is evaporation of a pure liquid. So here we have an example where we have petri dishes with pure ethanol. It's placed within a chamber in a grid, and then you place the chamber on top and measure for maybe half an hour, and then measure how much liquid you have left in the containers. And that should hopefully be kind of a homogeneous distribution. Another thing that's important, again, to validate your system is to see how much ammonia do you actually recover of what's emitted. And you can do this with some controlled test, for example, with recovery of ammonia emissions from a solution, where you know how much ammonium is in there, or you can have recovery of a standard gas through the system. And then, of course, we already know that these chambers cannot be used for absolute emissions, but we claim that they can give good estimates of relative effects. We, of course, also need to ensure that this is true, and we can do that by comparing with micrometeorological measurements. So here's one example where we compared emissions after application with trailing hose and injection, measured, and this is measured on the same plots, measured with the dynamic flux chambers, that's the red and purple lines, and compared it with BLS, which is a micrometeorological method, it's the green and orange line. In this case, we can see that the dynamic flux chambers clearly overestimates the emissions, but actually the relative differences between the trailing hose and injection measured with the dynamic flux chambers and the micrometeorological method BLS are comparable. And here's another example where we have measured flux with dynamic flux chambers, so DFC, and BLS, this micrometeorological method again, from the same plot, where we actually see kind of comparable emissions. So this is just to show that sometimes we overestimate with the chambers, sometimes we underestimate, and sometimes we get kind of the same results as with the micrometeorological methods. But regardless, we are really interested in the relative effects with the chambers. So a few other sources of error that one should keep in mind before you plan any experiments with dynamic flux chambers is whether you're going to apply the slurry by machine or by hand or like manually. A lot of research, also including my own, where we apply manure manually, mimicking applications by machine, because we can then use smaller plot areas. That's a big advantage. However, for some application methods, especially if there's an interaction between the application aggregate and the soil, it can be really, really hard to mimic it so it looks like the machine application, so that should be avoided. And generally, we have a lot of variance. I mean, nature is very variable, and a field is usually not very homogeneous when you go down to these small plots, for example, 0.5 square meter that a chamber will cover. So you really have to take into consideration how much of this natural variability you want to capture and how much you actually reduce the variability by choosing nice plots without weeds or things like this. And I don't think there's necessarily a right or wrong choice, but you have to bear in mind when you do the measurements, what do they actually represent? So I will also talk briefly about auxiliary data. So as you can see, this list is what I showed earlier with all these different parameters that affects the emissions. Ideally, we should quantify as many of these as possible and document them also when we report the emissions, because this can really help us to gain more knowledge about how all these parameters affect emissions. And this is just an example to show you why it is really important and we cannot rely on standard values. In the plot with all the points to your left, it's an example of samples I have collected for a project where we went to 9 biogas plants and selected this digestate effluent. And then we went to 3 commercial pig farm and cattle farms. They're all from Denmark, within the same region even. And you can see there's a large variability. So for example, dry matter for pig slurry, the 3 red points, we range from below 3 to above 6%. And if we don't know this, it can be hard to explain why we have differences in the emissions. And also to your left, you see an example of air temperature. This is from the same field a few weeks apart in Denmark, but where we tested the same methods, but saw different effects because we had a really big difference in the temperature within the first hours. That's the most important for ammonia emissions. So I know there will be a webinar later about modeling, but I just want to briefly talk about this ALFAM2 model that we use a lot for ammonia emissions. It's an ammonia loss from field applied manure model. First, I will mention that we have a database where researchers for many, many years have submitted their emission measurement data for ammonia. So right now there's data from more than 3,000 plots and it's all available online. Then the micrometeorological measurements from this database, the ones that we actually believe give good estimates of absolute emissions, is used to parameterize this ALFAM2 model. It's semi-empirical or semi-mechanistic. You can see how it looks here to your right, the picture you also saw earlier. And then the F's and the R's are the parameters that are estimated based on the empirical data in the database. So therefore, it's very, very important that we have as much data as possible. It's open source, so if anybody's interested, you can go to this webpage, alphan.dk, and there's an Excel version, and there's also an R version that is a little bit more advanced. But the Excel version is also good just to start to play around with. And why do I mention this? Because it's only parameterized with micrometeorological measurements. Well, it's to tell you that we have some current work in progress where we work on including chamber data, which would highly increase the number of plots used for the parameterization, and it should hopefully give a more accurate model. So, well, if any of you have worked with emissions of ammonia and have data to submit, you are very welcome to contact my colleague Sasha Hafner. He would be very happy to help you get in more data into the database. He's the one who's working with developing the model. So lastly, I will give some examples of recent research data. As I mentioned, we have a lot of slurry digestate in Denmark, and we have a challenge that we have a high pH and high dry matter in these, which both gives higher ammonia emissions. So we're really looking into how can we apply the slurry, or how can we reduce the emissions from the applied digestates. And we have looked at different application techniques. So to the bottom left, you see a trailing shoe, which is a low emission application technique in itself. But here we combine it with a hovering tine. That's what you see kind of hanging behind it that mixes the slurry and soil after application. And we saw a really good ammonia reduction by doing this. So we try to really lower the emissions during application. Other things we try is to do different treatments of slurry. And we in Denmark, we have worked a lot with separation of slurry. And generally, we see a good reduction in ammonia when we apply the liquid fraction compared to the unseparated slurry. But of course, one has to take care of the solid fraction, how to store and apply that as well. Another thing we do in Denmark is acidification, but usually quite high amounts are required. So in this project, we tried to use lower amounts of acid, but we actually didn't see a consistent reduction. But the point is here, we could do, because we have this automated system, we could do a lot of experiments with a lot of different digestates, and thereby also capture the natural variability, and therefore we'll have results that are more variable, but then our conclusions are more strong because we know we capture the variability. So this is my last slide. I'll thank you all for listening and say that we are open for collaboration, both me and my colleagues in our research group in the section of air quality and emission engineering. And just a small commercial here in the end, we have 2 PhD positions available. They're both on deep litter. One is about greenhouse gas emissions from deep litter, and another is from biogas utilization from deep litter. So if you're interested, please go and check out the calls. Thank you.
Thank you very much, Johanna, for a very nice and good overview about your measuring techniques and some of the outcome and also the modeling. So I will start the questioning. I hope there will be a lot of nice questions to you. Actually, it's a bit about mineral fertilizer. I'm sorry to switch to mineral fertilizers, but you told us so much about the manure. So do you think there is an option for actually develop a database with mineral fertilizers like we have with this ALFAM model?
Yes, I do think there's an option, and I think it would be very nice because it's something that could help us understand also the large variability we observe in emission factors from mineral fertilizers between different regions and management practices. I think it would be a big work, but I think it would be very important also.
Yeah. So maybe we should extend the manual management network to be a little bit more. Yeah, but I think that would be interesting. And then I have one more question. It's when you apply manure, if you wish to apply the manure in a plot and use and test, let's say, the big machines, then my impression is that you can weigh the machine with the manure before and after and get how much you spread. But it's very difficult to stop the spreading within the plot. So you're also spreading a little bit, or maybe quite a lot, unknown amounts outside your plot. How are you coping with that today? Do you understand
Yeah, I do. And for chamber measurements, that is not that important because you cover a smaller area. So you'll select an area that's within the well-defined plot or the plot where the manure has been applied properly. I think this is more challenge for the micrometeorological measurement methods where you'll have these unknown boundary effects.
Yes.
Yeah, I know some people have tried to cover the slurry in the outside the plot area with the plastic sheets, which I think can be quite challenging. And other people try to plow in the manure because then you really reduce emissions in that area. But it is difficult.
Okay, yeah.
So
you have a challenge actually also to calculate the application rate, I was thinking.
Yeah. I think actually the biggest challenge for the application rate for these small plot measurements like chambers is the slurry distribution over the slurry boom. So whether you have a 30-meter slurry boom or 3-meter plot trial boom, there might be quite large differences in exactly how much slurry leaves the different hoses if you have band application. And this should actually be quantified as well during your whole validation. Because you can have up to 40% differences. We measured that on a Danish machine a year ago. So that will, of course, highly affect your emissions if you then put your chamber on top of the area with 40% more manure compared to the other area.
Yeah, thank you very much. So they are opening for more questions. So I think you gave a very good presentation. I don't know if there are more questions. Otherwise, I have just
two questions in the Q&A.
It's coming now.
Yeah. So we have a question about the ALFAM2 model. From which region do the underlying data come? Are they mostly from Denmark, Europe, or are there also data from warmer region or even the global south?
That is a very, very good question. Most ammonia emission research has been conducted in the western part, so Denmark, Europe, Canada, US. So that's the major part of the data is from that, and that of course affects the model. So caution should be taken if you want to use the model in other regions. So, but this also comes back to the point that if any of you have data from some of these underrepresented regions, it's even more important that we can include that. So please, if anybody has it, feel free to contact me or Sasha Hafner so we can improve the model to better cover all regions.
And we have time for a last question. Did you see interaction with nitrous oxide emission with your CRDS measurements?
Do you mean interactions of, if we have a high emissions of ammonia than low emissions of nitrous oxide, or that the CRDS instrument itself can give interactions? If it's the last part, then the CRDS instrument I use, it's only for ammonia. It's not one of these greenhouse gas CRDSs that can measure both greenhouse gases and ammonia.
Great. So we still have 2 questions for Joanna, but because we need to move on to the other presentation, we will keep those questions and will be answered by email and distributed to the participants. Thanks, Joanna.
Thank you.
So now I'm coming back presenting the next speaker. It's Sven Sommer again talking. So the next speaker is Søren O. Petersen, who will give a presentation about chamber measurements of nitrous oxide emissions from arable soils, opportunities and limitations. And Søren is a very good friend of mine. We have been working together. So is Johan, I would say. We have been working together, and he is employed at the Department of Agroecology, Aarhus University. And in his research, he has focused on the interactions between soils, organic amendments, and microbial communities, with an emphasis on carbon and nitrogen cycling and the mitigation of greenhouse gas emissions in agriculture. He has much experience in measuring emission using a static chamber measuring technique. So Søren, the screen is yours.
Thank you, Sven. Hope this is the right one. Does it look right?
Yes, it's good.
Yeah. Okay, thank you very much. So thanks for the invitation and to present here and talk about chamber measurements of nitrous oxide emissions from arable soils. I'll say there are already a number of good guidelines out there on the use of chamber-based methods, and I'm not going to try and cover all these different aspects in this short presentation. Instead, I'll focus on some aspects that I find are important and some that I think it's important to discuss and that you may also want to have an opinion about if you are using static chambers. So there are many drivers of nitrous oxide emission. Of course, emissions are taking place at the microsite scale, but drivers of emissions, they are found at different scales. So there are some that are related to fertilizers, crop rows, perhaps. Some are related to field scale variability like topography, and other drivers are again related to soil types and climate. So they are at the regional scale. Some of this variation is systematic and easy to identify in the field. Others is more obscure or maybe even invisible and random and difficult to capture. And then there are, of course, also landscape features that could be taken into account when planning a measurement program. So how can we apply chambers in nitrous oxide research? So we have, of course, the well-known static chambers that we can use in field plots. We have automated chambers, and they come with different opportunities and limitations. But it is chamber-based methods is the method of choice to describe field scale variation due to soil conditions, unknown variation perhaps, and management. And this is also a point that Johanna made just before. And like Johanna, I'd like to say that it's really important to also collect supporting information on soil conditions in order to interpret what we see, and also to support later use of our data for modeling. And finally, I'd like to make just a note here that it's also really chamber-based methods can also be used under laboratory conditions and can be really valuable to examine the drivers and controlling factors in more detail. And we've done that quite a lot also. There are some potential risks that we need to consider when we determine the chamber design, selected chamber design. One is the disturbance of the crop. One is the temperature change that may occur during the measurement. And another one is the headspace mixing. That can also be an issue. I'm not saying that it's an issue here. I think in fact they tested and validated that this setup could work, but it's something to think about in planning studies. Also, the representativeness is important. So with a small chamber, small surface area, of course, you would need more replicates to achieve the same precision as you would with a larger chamber. And if you have crop rows or a band application of slurry or something, then you have, of course, some systematic variation that you can take into account by developing a chamber system, that chamber design that is appropriate for the systematic variability that we know is in the field. But that's something to consider. If you have a certain setup or you want to select a system for monitoring, there are a number of factors that are influencing what you can do with your system, and some of them are linked together. So, for example, if you have a large field experiment, you want to monitor nitrous oxide emission, then you have often a large distance between sampling positions. And if you're using a static chamber with gas sampling and GC analysis, then you need to walk far around to get to do your rounds for gas sampling. So that's induces maybe more non-linearity in the flux that you are observing. On the other hand, if you're lucky and have obtained a laser instruments, then portable laser, then you also bound to do measurements sequentially in the field, and that has other implications that we need to discuss. And finally, if you have a need to determine the high resolution of nitrous oxide emissions, then that also restricts the area that you can cover, the spatial variation that you can cover with your measurements. So the basic question when applying the chamber methods is how to best account for spatial and temporal variability, and what to consider when we plan studies. I have an example here to discuss the temporal variability, which can be very high for nitrous oxide, which is a very episodic type of emission often. So in this case, we applied the cattle slurry by injection to grassland in around 1st of June, and then we monitored nitrous oxide emissions with automated chambers for a month. You can see that some of these peaks were suddenly linked to rainfall, rain events, but then there was a tail of maybe 3-4 days before getting back to the background. Possibly the water was draining from the soil during this period. There were also cases where we had no emission, even though we had quite significant rainfall. And that's a warning that we need the information about soil conditions, because I do not know whether this is because we didn't have any nitrate in the soil at the time, whether the soil was very dry and didn't meet a threshold that would induce nitrous oxide emission, or maybe nitrate was leached from the soil, I don't know. So we need to understand this better. And then with the manual sampling, which many of us rely on, what is the effect of frequency of sampling frequency? And we use these data. Our colleague, statistician Marit Mainvey, used these data to analyze this. We had a 3-hour cycle, and she recalculated hourly fluxes based on this. And then she simulated manual sampling mid-morning around 10 o'clock with different sampling frequencies from daily, so 7 times per week, to 3 or 2 or 1 time per week. And then comparing that with the observed cumulative emissions as observed with the automated chambers. And she did 7 runs to vary the weekly sampling days. And these are the results. You can see the cumulative emission and then the relative bias. And you can see there's always a negative bias when we use manual sampling with the static chambers. We miss some emissions. Well, this bias was actually not that different on average between the different sampling frequencies. It's tended to increase with weekly sampling in this case. But you can see that the variability among the individual trial that she ran increased, so that with increasing sampling intervals, we also have an increasing risk that we get an estimate that it's deviating from the actual emission. So that's something to keep in mind. So it is inherently difficult to quantify nitrous oxide emissions with manual measurements. I wonder if treatment effects are more robust. That's something I really want to investigate, because if we sample from different treatments at the same time points, then maybe the ranking is similar, and therefore we would get a robust estimate still of treatment effects. Okay, so the calculated flux that represents the flux at the time of chamber deployment, so the time 0, we put our chamber on the ground. Does it make a difference whether fluxes are measured in parallel or sequentially? That's a question I'm asking myself, because increasingly lasers are replacing gas sampling for PC analysis in these years. If we look at soil temperature, in this case, I pulled temperature measurements from database. And the temperature at 5 centimeter steps on different time points, 9 o'clock, 10 o'clock, 11 o'clock, or 2 in the afternoon. Compare that with the daily average for each day in the year. You see that when we sample at around 9 or 10 o'clock, then the deviation from the daily mean temperature is quite moderate. Whereas if we sample later in the day, sometimes we have quite significant differences between the time points and the temperature and the daily mean temperature. And actually, for the data that we investigated here, the relative change in nitrous oxide flux was in the order of 5% per hour. So if you yourself your campaign with a laser takes 2 hours, then you might be looking at almost a 10% variation due to the deviation from the daily mean temperature if you want to represent the daily flux. So question is whether nitrous oxide flux is obtained sequentially should be corrected for a variable time of day, or if you can build it into the sampling design that you correct for or even out for this factor. That's something I would like to see discussion about. Another important aspect is whether nonlinearity is an issue. Based on theory, it can be stated that there is nonlinearity. Trace gas accumulation in a closed enclosure will be nonlinear by definition, because over time, when gases are emitted from the surface, there will be a saturation at some point in time. So I would say it's recommendable to allow for nonlinearity during flux calculation. So instead of opting for one particular flux calculation method, that you maybe allow for this flexibility. And I do want to highlight HMR, which many of you would have come across, which was the regression-based. It's a regression-based generalization of the Hutchinson motion method that was developed by my colleague Asger Rohr Pedersen. It's available in R as a package called HMR. And yeah, it has a lot of flexibility that I think is useful for this purpose. Theoretical background was described in this paper from 2010. Okay, here are some examples of flux measurements from a potato field on peat soil. So maybe nonlinearity is more pronounced here than it would be in other situations. But you can see that we have three plates with nitrous oxide fluxes on the left, CO2 fluxes on the right. And you can have nonlinear N2O flux and linear CO2 flux, or vice versa. You can also have linearity at one sampling day, and then nonlinearity on the next sampling day. So there's a lot of things we don't know about what causes the nonlinearity. But so that's why I believe it would be wise to allow for this to occur when it's the case. Another colleague, Rock Interior, he did a number of simulations in relation to one of the chapters for the guidelines that were published in 2020. He looked at precision and accuracy of different flux calculation methods, linear regressions, chronological regression, and HMR. And it's notable actually that when we have a short deployment time, at least with manual sampling, then linear regression has a higher precision and accuracy at low fluxes compared to the other methods. But it's striking that in fact, both precision and the accuracy of nonlinear methods increase with the time of chamber deployment. So if you have to leave your chamber on for a long time because you have a large field experiment, it takes time to get around in the field. It seems that this is in fact not an issue. You in fact capture the curvature better. That helps you to get a better estimate of the flux at time zero. Of course, when we extend the deployment time, also the risk that we are leaching losses, maybe from leakages in the chamber or the vent tube, or maybe horizontal gas transport in the soil would increase. It has been shown theoretically that leakage losses and horizontal gas transport are both the first order processes, and so they will affect the curvature, but not the flux at time zero when we use HMR. So that's also something that makes the method quite robust. I did a small simple simulation just in Excel to investigate the effect of leakage losses. And you can see here that so assuming 0.1% per minute or 1% per minute losses, we get different deviations from linearity. You see how linear regression would certainly have a strong underestimation by this, whereas HMR in fact maintains the estimate of the flux at time zero very well. It's not, there's a small change, but very small. So it seems to be a robust and quite reliable method under a variety of deployment conditions in the field. It's very flexible, but it's not very smart, this HMR. And so we need to consider also outliers. And if you have used HMR, you will know that sometimes, maybe by chance, if we have low or no emission, that the first observation is lower than the next observations, and you can have erroneous data like this one. And we need the methods to filter out these type of data. And there was some new features that was developed again by Astronaut Pedersen that were described in this, investigated and described in this paper. One is pre-filtering. So PFR is an option or function in HMR that you can use to filter the data so that you only allow for nonlinear flux calculation if you are quite sure that this is a real flux and not just random variation. And the filtered data, which are sometimes the most of them in a campaign, then I would really want to use linear regression because you cannot really determine whether there's a flux or no flux because it's like a gradual transition from no flux to very small fluxes and so on that we cannot identify. Another important function is this set time mean. So it determines how fast the curvature towards saturation goes and how fast you will allow this to happen. And this sort of constrains the curvature that you would allow, and that's based on experience. And in this case, this is one of the examples I showed before. It was in fact level off too fast for our experience, and it would be filtered out based on the criteria that we set. And the criteria for this saturation depends on the chamber design. How we now experience a large tall chamber would have a saturation time of at least 2 hours, and for the small dicor chambers in the front here, 30 minutes is more appropriate. Okay, so some opportunities for using chamber-based methods. Certainly for emission inventories, this is the method of choice. If you look at the IPCC guidelines, they are exclusively based on chamber-based methods. It is cost-effective way of studying management and effects of management and soil conditions, like we did in this particular study. You see in the field below, we have a 6-crop rotation with 3 replicates that we studied for 4 years in different locations. We have to acknowledge that there's a negative bias when we have infrequent sampling like we do with manual sampling methods. Perhaps we can use automated chambers. In this case, we had automated chambers in block number 1 to study the high time resolution. Maybe they can help us to estimate the bias. It's also an opportunity for using chamber-based methods to compare management practices. In this case, we studied 3 different nitrification inhibitors side by side for slurry and mineral fertilizers, and so that's also a very useful feature of chamber-based methods and allows for replication, so we can support statistical analysis of treatment effects. And with multi-site studies, we can also investigate interactions with site conditions. And in fact, this is something that we did in the last 4 or 5 years. And it can be useful also for model development if we have data from different sites. So emission factors are not really well suited to make predictions about site-specific emissions, for example, at farm level. And therefore, eventually, we will need to develop models that can help us to do that. And monitoring studies that we are doing should therefore be advanced so that it can also in the future support model calibration. And that could be supporting data on soil and climate, soil conditions, climate, and crop growth and yields and so on. And again, multi-site studies can support model calibration. And these are results from a 4-year study in 4 different sites where we used exactly the same fertilizers, manure, and mineral fertilizer. And we had different crop rotations that were representing the areas where some of the locations were distributed, and we monitored also ammonium nitrate, soil moisture, and so on, crop properties, and that creates quite a large data set that I think can be helpful in model calibration, and it is being used by colleagues now for that purpose. So some final remarks here. So chamber methods have great value for studying sources of variation, whether this is natural or related to management. there are some pitfalls related to the design and use of static chambers that I try to highlight some of them that I find are important. I see that portable analyzers are increasingly popular and replacing gas sampling with GC analysis. I try to highlight that there is a potential trade-off between the precision of the flux estimate, which is high with the laser, and the representation of daily flux. So it's something that I think we need to discuss more and find out how to deal with. And I would say that I think multi-site studies, they are really valuable for many reasons, and they may require a robust and low-cost method so that it's able to also cover remote locations with samplings, and therefore there may still be a role also for systems with manual gas sampling. Certainly, I'm not prepared to throw out our GCs at this time. I think they're really important, and we run 30, 40, 50,000 samples per year in our lab still for various studies. So there wasn't too much to livestock in this presentation, but they were there, always interested in what we were doing. Some acknowledgements. I really want to appreciate and highlight the contributions from Asger Roe-Pedersen and Rodney Venteria for the development of our knowledge about flux calculations methods and what we can learn from investigating fluxes. Marit Mengbe helped with statistical analysis for this presentation, and I have many colleagues from Aarhus University, from Marine University, and also the GIA Flux Guidelines team that I've interacted with over the years. So also thanks for their contributions. Okay, this was my last slide.
Thank you very much, Søren. I think there will come quite a lot of questions, but I have the opportunity to start, and that's this. We are looking in the Manual Management Network. We are looking to the south. It's called the Global South now, and very much into Africa and the need for measuring greenhouse gas emissions and ammonia emissions as well down there. So my question will be that I think they will have to use the manual sampling also because they shouldn't, and I know they will measure on many sites. So then the question is, at what time of the day should they carry out the measurements, and how can they define the best time? Because there will only be one sampling per day, I'm sure about that. So that is the one. And then how frequent, and should it be for 1 year, or is it okay to do it for 30 days? So that's the two questions I think I'll ask you, if you can advise us how to do it best.
Yeah, I think it's a good idea to investigate the diurnal variation in nitrous oxide emissions at the start of the study. But very often temperature is an important driver of diurnal variation. And if there was a review or meta-analysis of this a few years ago, I can send the link in the chat later on. And there are sometimes also nighttime maximum in emissions. But what we have seen is like a cycle where we have the two maximum during the day, one in mid-morning and one in the late evening. So I would say that mid-morning is still the best choice. So that would be my recommendation if you don't have prior knowledge about anything else, based on the temperature variation during the day. What was the other point that you mentioned?
Yeah, there's this discussion if for how long time should you measure? And in the past, IPCC said 100 days, but I've heard Klaus saying much more time.
Yeah, and I would say that you have to also consider the resources. I think it's important to document what is the annual variation. But what we have found in Northern Europe on light textured soils, that we have certain periods during the year, which is in the spring after fertilization, it's in the autumn after harvest and cultivation of the land, and also sometimes in winter. But in these are the three periods in the year where we see high emissions, and then there are other periods with lower emissions. So I think it's important to document the whole annual emission, but then also to focus maybe and make an extra effort, screen different fertilizer types or whatever in periods where emissions are high. It could also be cover crops that are killed by freezing temperatures in cold regions, like the northern regions. Also in Denmark, in some years, we have very high emissions during maybe December when we have the first real frost period, depending on the crop also. So we have to focus, make it balanced, so we can. We need to understand our systems, and then we can focus our efforts towards high emission periods after that.
Yeah, thank you very much. I'm sure there are other questions.
Yes, so Søren, one question is, this seems to focus on samples taken for GC analysis. How about measuring concentration in much higher time resolution with online instrument with recirculation? Sorry. Does the same sample apply with closure time and LR and non-LR?
Well, the theory behind is the same, but of course, with the short closure time, you would maybe see less nonlinearity, for example. I think I did discuss the use of laser-based methods in the field, and I'm just saying that there could still be differences in how the gas developed, and I think I would like to see some investigation into what assumptions about one or the other flux calculation methods influences this. And then I had this other point that we cannot measure with a portable laser at the same time in the different positions. And if it takes 2 or 3 hours to get around in a field experiment, then that could introduce an error, an uncertainty that we need to quantify and understand.
Right. We have a second question. A longer deployment time could result in a greater disturbance of the microenvironment being investigated. What precautions are being taken to reduce this disturbance, and how do they work?
So we use white chambers or reflecting chambers to avoid heating of the soil. But in fact, many of the disturbances that we observe, they will not influence the estimate of, or we have very little influence of the estimate of the flux, because what we estimate is the flux at time 0, so at the exact moment where we put the chamber on the ground. And so even if we have a small temperature change, if we affect the plant respiration, for example, if we have leakage losses, that's in the past it was really important to put your supporting frame 10 centimeters into the ground, and claiming that 2 centimeters could be enough because that may induce greater horizontal flux transport in the soil, but it will just influence the curvature, not the estimate of the initial flux. That's according to theory, and it's also according to the example I showed.
Thank you for that. So, and we have time, I think, for one question. So for manual measurement, how useful are estimation of diurnal fluid dynamics to assess how well the flux measurement represents actual fluxes? And how would you employ them if you were working in remote areas and had to choose your timing wisely?
I'm not quite sure I understood. Could you repeat the question?
Sure. How useful are estimation of diurnal flux dynamic to assess how well the flux measurement represents actual fluxes?
I think it's not something that you would want to do all the time. It's also really complicated to do, I suppose. But some understanding of, with a given soil type and climate conditions, how the diurnal variation is. But I think mid-morning sampling is quite a wise choice, and that's where it becomes an advantage to use manual chambers with gas sampling and GC analysis or subsequent analysis, because you can walk around and put the chambers on within maybe 30 or 45 minutes, and this is your time 0 that is constrained into this time interval. And then it doesn't really make a lot of difference whether your subsequent samplings take 1 hour or maybe 2 hours, which is the case sometimes for us.
Right. Thank you so much, Søren, for your presentation. Sven, the floor is yours again.
Thank you very much. So our last speaker today will be Dr. David Janke who will give a presentation about measuring, with the title "Measuring Gaseous Emissions from Livestock Housing System with Tracer Gases." So it's the tracer gases that are in focus, David. I know that. Well, so David Janke has specialized in measuring emissions, mainly methane and ammonia from livestock housing systems. So his focus is on the coupling of different methodological approaches in order to develop high scalable, lower cost measuring routines. So the floor or the screen is yours, and I look much forward to learn more about the tracer gas method, actually. Thank you very much for coming here.
Thank you very much, Sven, for the nice introduction. So I would just start. If you don't hear me well, just give me a sign and I try to fix something. Talk today,
I can
measuring
hear you.
gaseous emissions. Very good. From livestock housing systems with tracer gas methods. And just to set the picture for the next 20 minutes, what's it all about? System boundaries. I think it might be important in this context. We're moving now from the fields or field application to barns for livestock. And with barns, usually I mean housing systems that are naturally ventilated, either for pigs or cattle, also maybe in the future a bit for poultry. And I will make a little excursus also to external slurry storage systems. The pollutants I am mainly interested in are anything that is airborne leaving the barn, which we do not want to have in the environment. Can be ammonia, CH4, nitrous oxide, PM, and any other stuff that leaves with the airflow of the barn. Today, I'll focus on ammonia and methane. And today I talk really about methodology. I will not show any emission factors for certain husbandry systems or really absolute values for the emissions of any system. Yeah, after the session, hopefully you'll have understanding of how we measure emissions from livestock housing systems at the moment, what we're really interested in the current state of research, and most important from my point of view, the associated limitations and not so often communicated uncertainties, talking about errors. So let's see. I structured the following talk according to 3 questions. First is, how do we measure? With a little theoretic part. Then what is the achievable accuracy when we measure? And very interesting in the end, what are the current research questions? So where are hot topics where we can still learn very much from? So let's start with theory. Many of you will, of course, know this, but I try to really start from the beginning. The emission itself is a mass flow. So usually it's measured in kilogram per second or gram per second. And emissions are nothing more than a volume flow. So a moving air, which is cubical meters per second, that transport any kind of pollutant, which is usually given in a concentration, which could be kilogram per cubical meters. If you have a look at the units, this equals to kilogram per second, the mass flow, the emissions. And if we want to measure emissions, we need to be able to measure the volume flow and the concentration. And volume flow, I try to picture it here as the blue arrow. So this is anything where the air leaves a certain specified area at the system boundary with a certain velocity. And it transports the pollutants, which are shown here in the yellow pixels. These yellow pixels are the concentrations which we can measure usually very accurately. We heard before in the talk of Johanna already some measurement devices. Usually we use Picaro, so it's CRDS spectrometers or FTIR. So this is a problem which is not really a problem. We can measure this easily. When it gets to the volume flow, it can be a bit more complicated. If we are having a look for mechanically ventilated buildings or housing systems, this is not problematic. So we have a green thumb here, no problem at all, because we can just measure here the ventilators and would have then the volume flow. But as I said before, we are here in the world of naturally ventilated buildings, and this is where usually the challenges begin. Our sources are inside these buildings, and they are very large, and they usually have several thousands of square meters of opening. And followed by the wind, they emit airborne pollutants. And this always results in a super complex flow pattern. I try to picture this here with a sketch. So we have the flow itself, we have the interaction with the building, we have different climatic conditions, we have different animals. So from a measurement perspective, usually it's a mess. And yeah, saying in short, the measurement of volume flow Q is more or less complicated. This is why we do not really measure it directly with anemometers. We usually go another way, and this is the tracer ratio approach. You see here the equation E equals Q times C. I can easily morph it into the Q equals E divided by C. So this for us means I can describe the volume flow as the emission, which is the mass flow, divided by the concentration. Okay. And if I am not on the emission side now for the pollutant gas I want to measure, but if I imagine a tracer gas, so a gas which I myself put into a system with a known mass flow and measure the concentration in the system of this tracer gas, I get the Q on the left side, I get the volume flow. And this is basically the main idea of tracer ratio approaches or tracer gas methods. I make it here at the example of the emission of methane. So if I want to measure methane, I have to measure the volume flow and the concentration of methane in the system. And I just replace the Q with the equation I have here with the mass flow of a tracer gas divided by the concentration of a tracer gas times the concentration of the pollutant gas, which in this case is the CH4 concentration. And that's actually the main idea of doing so. So in a picture, we have here the system. We have a source of the emission inside the barn, which is methane. Then I put in the tracer gas and release it with a known mass flow. So the M I know. And then all I have to do is just to measure most you know what I mean? So it must be representative and measure here the concentration of CH4 and the tracer gas at the same time and also at the same location. And that's actually everything I need to do. So that's the basic idea of applying tracer gases. And then you have, of course, when you put this into action, you have to have different decisions. First would be what's the choice of the tracer gas. Actually, we can divide between two tracer gases. First would be artificial, and the second would be natural. We start with the artificial tracer gas. In most cases, as tracer gas for artificial applications, so something you really inject into the barn with a mass flow controller would be sulfur hexafluoride, in short SF6. This is a chemically inert and non-reactive gas, which is actually not really measurable in the ambient background concentration, which is good because you have a really great signal-to-noise ratio. It's detectable at extremely low concentrations, which means you do not have to put in so much gas into your system, and it's stable over time. These are all very good things, and this is why this gas is taken as the one gas for the artificial tracer gas injections, but it has several negative aspects. So usually it's measured with a GC ECD, and this is mostly very cost intensive, and you have to have really knowledge of the system, and you cannot measure the pollutant gas with the same instrument you use for the tracer gas. You need to install an injection system with mass flow controllers, and most important, it has a very high global warming potential. So this is a really very, very potent climate gas. So actually, it's good to measure, but there are many concerns to further use it for experiments in such high concentrations. Giving a short sum up on the artificial tracer gas SF6, these techniques are considered among the most accurate methods because you really know what you put into the barn and you can measure it very correctly, even at low concentrations. Drawbacks, as said, is expensive. You can use it only for short terms because of the high GWP. So Usually, alternatives to this artificial tracer gas are applied. This leads me to natural tracer gases. Most commonly used as a natural tracer gas is the carbon dioxide CO2, which is produced by the animal metabolisms inside the barn. This has several advantages. So you do not need an extra injection system. You really have a good homogeneous distribution of the barn because the cows are always moving around. And also very good, the tracer gas and the pollutant gas can be measured with the same instrument in case you have a good working CRDS or an FTIR. This is all good. Yeah, the bad things are especially that the mass flow of the CO2. We do not have a mass flow controller in the cow where we can really define the mass flow. But we try to model it by the production term, which we model according to the properties of the cow, like the mass of the cow, days of pregnancy. I'll come to that later. So this is a source of error itself. And also the inside concentrations measures have to be corrected for ambient outside CO2 concentrations that come into the barn. This has a really large influence where you put your measurements. So these are definitely drawbacks, and I come to this later also when we have a look at the errors that are inherent to this method. CO2 tracer methods are most commonly applied approaches. Nearly all the papers you find for measuring emissions from livestock housing will use these methods because it's easy to apply. Drawbacks, obviously, you have more error sources than using artificial tracer gas SF6. Yeah, practical application of this method. You have to set up a measurement setup inside the barn and around the barn where you continuously sample the air inside the barn, and you have to continuously sample the air entering, so outside the barn. You bring this air to your measurement devices, either CRDS or FTIR, and the aim is really to have a representative sample of the leaving concentrations. And this is somehow very complicated, and you have to put much work of it. You see here the red dots. These are all openings from measurement lines. So these are sampling tubes made of PTFE, which we install inside every barn. Sometimes they are nearly 1 kilometer only sampling tubes you have to install. And you can imagine how long this takes and how expensive this is. Usually you have to measure, yeah, at least if you want to have really emission values for the year, at least. So this is the absolute minimum, 6 weeks distributed over the year. Yeah, what we do is we usually measure one emission value per hour at least, and we give per sampling locations 7 minutes of measurement to have stable results per sampling location. And this leads to the preliminary conclusion that even with cheap CO2 tracer methods, measurements are very cost and resource intensive. And this leads to that we really cannot measure at the moment the large amount of barns which we should measure in order to really have a picture on the large variability of all the barns and all the systems. Not even in Germany, but also in whole Europe and all over the world. This brings me to the next question. What is the achievable accuracy? And here I would like to focus on CO2 and go on with the naming just some error sources. The first and probably most important would be the CO2 production term. As said, we model this, we cannot measure it. So it's based on some assumptions on the heat production of individual animals that were achieved with respiration chamber experiments, and it takes into account several variables like the days of pregnancy, the milk yield, the mass, the temperature, the activity of the cows, and so on. And it's mainly based on older experiments and literature review. The most actual was in 2008. And of course, there are concerns because the cows developed over the years, and new studies from Wageningen University indicate that the use data might be outdated, and we have systematic error because, of course, the metabolism of cows now compared to nearly 20 years ago is a bit different, and they have developed. Second error source potentially for the CO2 production term is the portion of CO2 from manure which is stored in the barn itself. This produces CO2 as well, but this is still very unclear how the portion of the metabolically produced CO2 from the cows is compared to the CO2 produced from the slurry. And roundabout, you can expect here errors in the range of 15%, probably a bit more, but data is not very clear here at the moment. Then another error source, very important, and yeah, still yeah, in the focus of our research is the sampling method itself, spatially and temporally. It yeah, can be concluded to the question, where should we measure the inside concentrations and where should we measure the outside concentrations? If you have a look at the picture up right, you see here 6 different possibilities in red where you could measure with a sampling line the concentrations inside the barn. And of course, you could do the same for the outside. You could measure west, north, east, or south. And then you just have to draw a decision. Okay, this is my line, and here I put it. And this decision is very meaningful for the emission estimates. So we found that we could gain up to 26% of error only by choosing the position of the sampling line. And this is usually a systematic error. If we have a look at the duration of the measurement, you can have a look at the picture down on the right. So this is for the air change rate, which is directly connected to the volume flow, and this will have a direct influence of the emissions. And here you see the influence of the measurement days on the confidence interval, and you see how the random error arises when you just reduce the days of measurement. So both is super important and also pronounced to errors. First, the location of measurements and also the duration of measurements. And again, this number is around about errors up to 26% possible. If you do it really in a very wrong way, I imagine the errors can also be higher. Here it's really the achievable accuracy if we assume that the SF6 tracer method delivers the true or the right value. This was or is still an interesting study from the colleagues of Agroscope in Switzerland. They made a direct comparison, or at the moment analyzed a direct comparison of CO2 and the SF6 method. And these were measurements done over winter transition and summertime. And we just compare the emission values for CH4 in this example for a barn with a for a barn with a plain floor. And the results after having a first look were surprisingly good. So what you see here in the orange dots, this is the SF6 method. What you see in the blue dots is the applied CO2 measurement methods. And the average deviations over the whole winter-summer transition was less than 20%. And yeah, here we were a bit surprised because we expected way worse. And this made us yeah, happy because the results are really promising for the reliability of CO2 as a tracer. Going on with some positive news. Here we had the interest in the achievable precision of the CO2 tracer method. With precision, I just mean what is the repeatability if different measurement institutes go at the same barn, set up their own equipment, and measure their own way, following the rules of the CO2 balance method. And we made a round-robin test at our experimental barn. We had 5 different institutes that measured in parallel the same barn. We measured emissions of CH4 and NH3. And yes, what you can see here is, in a nutshell, we couldn't find any significant differences for the emission estimates from each institute. I don't want to go into detail for the two pictures. Bottom line is, from a statistic point of view, no difference could be found. And this was really very promising for us because in a larger project, we would like to use the CO2 balance method as the reference value, and then it's good to know that it's at least applicable with a good repeatability. Coming to my last point, the current research questions were a hot topic at the moment. And this is just a overview of some actions we do in a larger national joint project called EMIMOD. Here, our colleagues are trying to develop a reference method for external slurry storage. They are investigating different methods, and as the, let's say, true value, they apply SF6 as a tracer, and they let it release at the surface of slurry tank. And downwind, they measure then the to be measured pollutant gas and also the tracer gas. And by this, they deliver values for different models and also micrometrological methods, flux methods, in order to have a validation data set to assess the applied methods. This is, as said, ongoing. I can't show any results so far, but pretty soon, I guess, we can publish here something. Current research is also, this is super important for us, measurements of exercise yards, all for cattle, pigs, and poultry. We're doing experiments for the simplification of the SF6 tracer ratio, where we don't need to have the whole setup of injection lines and this whole mass of SF6, but trying to put on reduced order models. We try to also develop these methods then also for extremely complicated conditions like exercise yards for pigs, but also very important in the future for us are these exercise yards for poultry. You see on the left the picture for a winter garden, and this will be very important in the next 1-2 years in our research. And also this is the framework of the ongoing national project EMIMOD. And this already leads me to my summary and conclusions. If we are looking for emission measurements in livestock, usually we apply indirect tracer ratio methods, either with the artificial tracer gas SF6 or the natural tracer gas CO2. It's very demanding and expensive. We still have relatively high uncertainties inherent to the method. The analytic itself, so the measurement device, is not the main concern. It's rather the methodology and how you can reduce the uncertainty here. And if ever you see results from measurements like this, always have in mind we have high uncertainty ranges. And for example, if I use a tracer ratio method in order to assess a mitigation method that brings maybe a mitigation of 10%, keep in mind the measurement method itself might be not as accurate as your proposed mitigation potential. For the future, we see very exciting challenges in the measurements of exercise yards, winter gardens, stuff like that. In the development of less expensive methods in order to have more scalability and take into account the high variability in between farms. And a very important topic is the alternative for an artificial tracer gas. So it doesn't have to be SF6. There are other gases that might also be interesting. And with this, I thank you for your attention. We're always open for collaboration. Just reach out. I put my email here, or maybe we can have in the discussion right now.
Thank you very much, David, for a very, very nice and informative presentation. I had a lot of questions, but you answered them before I could ask you now. So I just have one left, and that is, or maybe two. One is this CO2 from the manure itself. How much do you think that will affect the emissions? And my second one is we like to have a more focus on the global south, and if you go to Africa, where there are a few measurements from, then you have the pastoralists. They have their animals in yards like your winter garden, or could you use this CO2 technique there? So So that's my question. So could you really, is it possible to use it also, in a country where you don't have too much equipment or infrastructure?
Maybe to the first question, the portion of CO2 produced by manure, this, as always, depends. It's usually a rule of thumb, you can put something like 10 till 15%. It's still unclear, but I know there are very detailed studies ongoing at Wageningen University, and probably I think they are about to publish this because they can measure the portion of the manure itself without the animals, which we cannot do. But I'm not aware of already published data at the moment. So sorry, I cannot answer this. We use it more as a roughly rule of thumb. And of course, it depends how long the manure is in the barn. Is it stored in the barn very prominently or not? These are questions we do not tackle at the moment. And the second question, at the exercise yard, it will be very hard to measure. And we already have some contacts to Benin in Africa, where we aim to also do some measurements in animal housing systems, but not with a conventional measurement setup, but with a low-cost device which is equipped with electrochemical sensors for ammonia and NDIR sensors for the CO2, and this can be just sent to there and it can be installed. Of course, the precision wouldn't be the same. For exercise yards, I'm very skeptical this would work if you do not really have the instruments, and this would include also have technicians there who can maintain the instruments probably 24/7. But then I believe it will be possible, yes.
Thank you very much. So there are more questions, I'm sure.
Yes, David, so how should 15-20% method inherent uncertainty in tracer-based measurement be explicitly handled in GHG methodologies to balance integrity and feasibility?
Could you repeat? I didn't understand
sure.
it quite well.
How should 15-20% method inherent uncertainty in tracer-based measurement be explicitly handled in GHG methodologies to balance integrity and feasibility?
How we should handle this uncertainty?
Yes.
Yeah, we always have to communicate, and it's always important for what you use the method. As example, if you use the method at the same location in order to identify relative changes, you have a baseline which you measure with the method, then you put in a method for mitigation, you measure with the same method, then this is not really such a dramatic thing if you have a systematic error of the method. If you use the method for measuring emission factors for inventories, this is what we always communicate. We need to make sure everybody understands this is not one value. This value is really associated with uncertainty, and you just have to communicate it.
Thanks, David. And I have one question. So you mentioned that we should measure in many bands. Do you have any idea about the variation between bands compared to between periods? As it is a very intensive and expensive work, would it be better measure in more different bands for each time?
Yeah, definitely. So we saw variations. We had a larger measurement project where we measured with this method, really with high cost instruments for each system variation in 4 barns, and the variabilities, they were awful. So you couldn't drive or you couldn't put on any statistic that would make sense. Even the temperature dependency was not clear if you put all the data from all barns together. This just means that the intervariability of housing systems, and even if it's the same housing system with the same management system, is so large that you can tackle this only if you increase the number of investigated systems. And obviously, you cannot do this if the methods are so expensive. So one of our large research paths is to develop methods that have nearly the same accuracy, but are maybe a tenth of the cost which you have at the moment.
Thank you so much, David, and all the speakers. Sven, back to you for the closing remarks.
Yes, thank you very much to the three speakers. I really enjoyed your presentations and I learned a lot. So that's marvelous. And just a few final comments. It's that as the audience might have heard, that all of you are open for questions and contacts, and if people need something for to get started and to measure correctly, then they can contact you. And if they don't have your email addresses, then they can contact the Manual Management Network or me, and I will send them your email contact information. And then I would also say in April we will have a webinar following up on these webinars we have had until now about measuring techniques, and that webinar will be about how to develop models or 3 different methods for developing models and model outlines that will, from the IPCC and from colleagues, will be presented in April. We haven't fixed the date now, but there will be a webinar by then. And again, if somebody in the audience or colleagues to the audience are interested, then we are open for organizing webinars where that is organized by you. So thank you very much for listening, all of you, and a great thank to the 3 presenters for very nice presentation and well organized. Yeah, thank you very much.